Aziz Sancar was born in Turkey in a small town called Savur in 1946, same year as this previous speaker. He moved to the United States after having been a medical doctor in Turkey. He went to the United States and got his PhD at the University of Texas in Dallas. And he's now Sarah Graham Keenan Professor of Biochemistry and Biophysics at University of North Carolina at Chapel Hill. Welcome to the stage, Professor Sancar.
Thank you very much. I'm truly honored to be here, and I'm grateful to the Nobel Prize in Chemistry Selection Committee for recognizing our work on mechanism of DNA repair by photolyase and by nucleotide excision repair system, which I'll present here. I think my presentation is much longer than the first two, and I do want to make a point because I don't want to get into a rush in the end. It is an important point. Dr. Lindahl mentioned yesterday that science research is not done in vacuum. We all built on research done by our predecessors as well as our contemporaries who have contributed in significant ways to our own research, to my personal research that I will present here. But I will present data on photolyase and excision repair. These are two DNA repair systems. And I will also present data on circadian clock, which connect these two repair systems. But I really want to emphasize the contribution of many scientists in all these areas that have been instrumental in accomplishing what we have. And I just did not want to leave it to the very end so that it's mentioned in a hurry and without proper recognition. So I am grateful to all these colleagues who over the years have contributed the necessary both intellectual background and technology and frequently reagents that has enabled us to carry our own research. With that said, then I will present to you our work in these repair fields. I will talk about two repair systems, photolyase and nucleotide excision repairs, and these two repair systems repair DNA damage by ultraviolet light. Ultraviolet light acts on the ultraviolet light component of sunlight actually acts on two adjacent thymines in DNA and converts them to a thymine dimer or cyclobutane pyrimidine dimer, which is mutagenic, carcinogenic, and lethal to the organism. And this damage is repaired in E. coli by an enzyme called photolyase, and in E. coli and humans, by an enzyme system called nucleotide excision repair system, as shown here. I will conclude my talk about how our research on photolyase led to the discovery of an essential clock protein called cryptochrome that linked these two research subjects, meaning photolyase and nucleotide excision repair, to one another, and therefore completed the circle. Photolyase is what you may call a photon-powered nano machine. Nano is popular now. Everything is nano, and photolyase is a nano machine that uses photon to repair thymine dimer that is induced by ultraviolet light. Photolyase was discovered by my mentor Stan Rupert in 1958, and the discovery of photolyase is considered the landmark discovery that established DNA repair as a scientific research topic. Decades before this discovery of photolyase by Dr. Rupert, it was known that ultraviolet killed bacterial cells, like shown here, depending on the dose, with great efficiency, and that was repeated many times. But in 1949, Kellner from Brandeis made the interesting observation that if bacteria killed by ultraviolet light were exposed to blue light, miraculously they were brought back from the dead. And that was a miraculous finding, of course, and he had no explanation for it. Then Dr. Rupert took this issue and analyzed it, and what he did was, first, he demonstrated that ultraviolet light kills cells by damaging DNA, and that there was an enzyme that repairs the DNA damage in a light-dependent manner, and that was the reason for resurrecting light, resurrecting dead bacteria from light. There was no miracle, there was no metaphysics in it. It was simple physics. And the reaction mechanism that Dr. Rupert came up with is summarized here. Ultraviolet converts two adjacent thymines to thymine dimer, and there is an enzyme called photolyase, which uses blue light energy to break these abnormal bonds and convert the thymine dimer to two normal bases, and therefore repair DNA and eliminate the harmful effect of ultraviolet light. Now, while this was a satisfactory explanation, it raised a physical problem. Photolyase is an enzyme, it's protein. Proteins do not absorb blue light. And therefore, for the next 2 decades, Rupert and many other scientists tried to identify the blue light absorbing component in photolyase, and they were unsuccessful because Dr. Rupert had determined that an E. coli cell contains only about 10 to 20 molecules of enzyme, and this proved impossible to purify. So in 1974, when I joined Dr. Rupert's lab, the gene cloning had just been developed at Stanford, and as a fresh graduate student, I thought I could do anything I wanted, and I proposed to Dr. Rupert to clone the photolysis gene and overproduce it and purify it, and he said, go ahead, and I cloned the I cloned the gene, and this is the plasmid with the photolysis gene on it. And in the subsequent years at the University of North Carolina, my colleagues and I purified this photolysed protein in gram quantities. And while we purified it, we found that it has a bright blue color. And right there, that finding, without any analytical chemistry, answered the question of what was it in photolysis that absorbed light and catalyzed reaction. It had a blue color, meaning it absorbs light. Nevertheless, we had to identify what the material was that absorbed blue color, and we carried out analytical chemistry analysis on the enzyme, and to our surprise, we found that it contained not one, but two cofactors, one flavin adenine dinucleotide and the other folate, and that the enzyme actually could exhibit colors ranging from purple all the way to orange, depending on the various redox status of the flavin cofactor. In any event, the enzyme contains two cofactors. One is folate and one is flavin. And our next task was then to determine the function of these two cofactors. By carrying photochemical experiment, we found that folate actually acts as a solar panel. It absorbs light, and just like solar panel, it transfers the energy to the flavin, and the flavin is the catalyst that carry out the catalytic reaction to split the pyrimidine dimer. To provide structural basis for this photochemical data, we collaborated with Hans Deisenhofer to obtain crystallized photolysis and obtain a three-dimensional structure, which is shown here in ribbon diagram and a surface chart representation. And like I said, folate is really a solar receptor, sits on the roof of the protein, gathers blue light energy, and transfers the energy to the flavin in the core of the protein to carry out catalysis. With this general structural view, then the mechanism for photolysis was developed as shown here. The enzyme binds to DNA containing thymine dimer, because thymine dimer causes abnormal structure in the backbone, and it pulls the thymine dimer from within the helix into van der Waals contact with the flavin, and it makes a very stable complex. Nothing happens until a blue light photon is absorbed by the folate, which transfers the excitation energy to flavin, and the flavin repairs the cyclobutane dimer or thymine dimer by a cyclic redox reaction. The dimer is repaired, and the enzyme dissociates from DNA and goes on to search for new damage site and to reinitiate the cycle. Over the past 10 years, in collaboration with Dongping Zhang from Ohio State University, we have determine the microscopic rate reaction for every single step in catalysis, from absorbent light, energy transfer, and various steps of electron transfer, bond cleavage, bond formation, and back electron transfer. To make a long story short, we have the rate constant at picosecond resolution in real time for the catalytic reaction, and the entire catalytic cycle takes about 1.2 nanosecond. Photolyze, in addition to its intrinsic value, is actually contribute to the other major repair mechanism that's found in many organisms, and that is nucleotide excision repair, which is the other topic of my research. In the early days of work on Photolyze, the way the experiment was done, cells would be responded in buffer, and they would be irradiated with ultraviolet light, and half of the sample would be kept in dark, and the other would be exposed to blue light, and then they would be analyzed for thymine dimer content. Of course, the one exposed to blue light, thymine dimers would disappear. The one that was in dark, thymine dimers repaired in the genome. However, if a small change was made in that design, if the buffer contained glucose, it was found that even the ones that were kept in dark lost the dimers from the genome. But there was a difference between that loss and the loss that was caused by photolysis. When the cells were kept in dark in the presence of glucose, even though the dimers were lost from the genomes, they were within the cells. They were gone from the genome, but they were in the cytosol. And that led to the concept of nucleotide excision repair by Howard Flanders at Yale and Settler at Oak Ridge National Laboratory. And that is the next topic that I'll be talking about. So after this initial discovery by Settler and Howard Flanders, research was done in many labs from 1964 till 1982. And the basic conclusion from these studies are summarized here. Thymine dimers are removed from the genome in both E. coli and humans, and excised thymine dimers were found to be in oligonucleotides, 4 to 6 nucleotides in length. And it was also found that this repair reaction is genetically controlled by the UVR genes in E. coli, UVR A, B, and C, and XP genes in humans. And XP genes, there were 7 of them, XPA through XPG. And that the basic reaction mechanism is what we call endonuclease, exonuclease coupled with repair synthesis. But importantly, the excised dimers remained in the cell as opposed to photoreactivation. So this was the view in 1982. And in 1987, after my initial work on Photolyze in Dr. Rupert's lab, I joined Dean Rupert's lab at Yale University to study the E. coli nucleotide excision repair. And for that, I cloned the three genes that were implicated in excision repair: UVRA, UVRB, and UVRC. Nothing was known about these genes. And what they did, I developed the MaxiCell method for specifically radiolabeling plasmid encoded gene, and identified these three proteins as 100, 85, and 70 kilodalton proteins, and then purified them in milligram quantities as shown here in a Kumasi blue stain gel, and asked the question, how do these proteins carry the repair reaction? And what I found was this, and this was different from the classic endonuclease-exonuclease reaction mechanism, in that the three proteins acting together made incisions at precise site, 7 nucleotides 5' to damage, and 3 nucleotides 3' to damage, to release an oligonucleotide of 12 nucleotides, and then the gap was filled and ligated. And having the basic mechanism is established with these three proteins, then later on at the University of North Carolina, my colleagues and I studied the individual function of these proteins, and we established this reaction mechanism. UVRA recognized damage, recruits UVRB, promoting a very stable UVRB damaged DNA complex, which then recruits UVRC, has the 2 nuclease active site, makes the dual incision, and then UVRC is displaced by helicase, UVRB by polymerase, and the gap is filled and ligated. While this work was going on, Phil Hanawalt at Stanford published a paper showing that both in E. coli and in humans, transcription strongly stimulated repair. And he and others suggested that the reason for this was because the rate-limiting step in repair is the damage recognition step. And the explanation was that RNA polymerase stops at the damage site, cannot pass the damage, and it constitutes a large target, and as a consequence, it is a good target for damage recognition, and that's how it facilitates repair. So we tested this model in a defined system, meaning the UVR ABC and RNA polymerase with substrate-containing thymine dimer. We found that in contrast to that model, stalled RNA polymerase did not stimulate repair, but actually inhibit it. And therefore, we proposed that there must be a protein that removes stalled RNA polymerase, and on top of that, recruits the repair protein to accelerate the repair rate. And we proceeded to purify this protein using biochemical assay, and the results are shown here. We purified this protein. We called it transcription repair coupling factor. Turned out that to be a gene that had been identified decades ago with regard to mutagenesis study by Evelyn Witkin, and was named MFD. In any event, MFD, or transcription repair coupling factor, indeed recognized stalled RNA polymerase at damage site, displaces it, and while concomitantly recruiting UVRA to accelerate the repair rate. So at this point, we figured that we had completed the description of prokaryotic repair, though in science never anything completely complete. But we moved on to study human excision repair. And that is what I will present to you in the next few slides. In human excision repair, as I mentioned, it was found that, like in E. coli, human excision repair basically proceeded by same mechanism, meaning 5' endonuclease recognizing damage to make incision, and then an exonuclease removes the damage in 4 to 6 nucleate long oligomers, concomitant with repair synthesis to filling the gap. So this was the current state of knowledge when we started working on nucleotide excision repair. In humans, I would also point out that in 1968, James Cleaver had found that humans defective in nucleotide excision repair, or rather patients with very light-sensitive disease called xeroderma pigmentosum, were defective in nucleotide excision repair, and moreover that there were 7 genes that were responsible for this disease, meaning mutation in any of these genes could cause defect in nucleotide excision repair and therefore xeroderma pigmentosum. And I will show you a picture of a patient with xeroderma pigmentosum. Some colleagues found the picture very disturbing, and they advised me not to show it more than 5 seconds, and that's what I will do. It is very severe disease. Patients have 5,000-fold increase in sunlight-induced skin cancer, and I will not keep it more than 5 seconds. So using our expertise in working with E. coli excision repair, we went on to characterize human excision repair. First, from the very beginning, we found that the human excision repair, just like E. coli excision repair, is also carried out by dual incision. But beyond that, it was very different. To begin with, in human excision repair, not just 3 proteins, but in fact 16 proteins and 6 repair factors shown here were absolutely essential for making the dual incision. And furthermore, these proteins are evolutionarily not related to the excision repair proteins involved in prokaryotic repair. Secondly, even though in principle both E. coli and humans carry out excision repair by dual incision, that's where the similarity stops. The dual incision mechanisms are quite different, whereas in humans, the incision is 7 nucleotides away from the damage. In humans, it's 20 to 22 nucleotides, 5' to damage, and 5 nucleotides, 3' to the thymine dimer. As a consequence, humans remove thymine dimers in the form of a 30 nucleotide long oligomer. The difference does not stop here. In actual mechanism, also the damage recognition and processing is quite different. The damage is recognized by these factor XPR, PXPC, which recruit TF2H. TF2H as the helicase ATPase unwind the helix, and then recruits the 2 nucleases, XPG and XPF, which makes the 5', 3' incision, and then the repair is completed by polymerase and ligase. Now, in all these studies over the years, both us and other groups working on human nucleotide excision repair, this was confirmed by many other groups that humans did excise a 30 nucleotide long oligomer, but really a serious issue remained, and that was many studies prior had published that humans excise the oligo in 4 to 6 nucleotide long fragments. And here, all these experiments were done in vitro, and we identify a 30 nucleotide long fragment in the in vitro experiment. And therefore, there was a discrepancy between in vitro and in vivo data. And it took us 21 years to resolve this discrepancy. And the key was we found that the excised oligonucleotide, the 30-mer, is in a very tight complex with this repair factor called TF2H. Once we found that, then we said we can immunoprecipitate it and measure the actual size. When we did, it indeed turned out to be in vivo also 30 nucleotide, and that discrepancy was because in the other in vivo experiments, the oligo was degraded. In any event, in vivo, the oligonucleotide is also 30 nucleotide in length. But this also provides us the means of generating a repair map for the entire human genome, as shown here. We irradiate cells with UV, photoproducts are made, and the dual incision release 30-mer, and that's in a complex with TF2H, and we immunoprecipitate TF2H to isolate both TF2H and the oligonucleotide with it, then extract the oligonucleotide and sequence it by next generation sequencing to generate sequences from the entire genome. In a typical experiment, we get 20 million sequences. Then we blast these 20 million oligos against the human genome to generate an entire map for a repair map for the human genome. And this is an entire map for the human repair genome for a male individual of 22 plus X plus Y chromosome. The black tracks show the transcription tracks, and the green are repair tracks for two types of photoproducts. Of course, this is a screenshot of the entire genome, and you cannot really tell much detail of what's going on. I will concentrate on one specific chromosome, 17. This is an important chromosome. It contains the P53 gene, which is mutated in about half of human cancer, and that will give a better idea of kind of information one can get from the human genome repair map. This is chromosome 17, is 83 megabase, and here we're showing the transcription of plus strand and minus strand, and the excision repair, we call it XR-seq, excision repair map for the whole genome. And it is really a map in the true sense of the word, in the geographic sense, meaning it has mountain, it has valleys, and it has canyons, meaning high repair, low repair, and no repair at all. But importantly, we can ask question for any particular position, nucleotide or dinucleotide, how that place is repaired. And just to give you an example, this is the location of P53 and the location of P53 dinucleotide, which is highly mutated in skin cancers. So at megabase, that's where it is. At kilobase resolution, this is the P53 transcript map, and this is the P53 repair map. The specific hot spot for UV in this cancer is this dinucleotide, and here our repair map shows how this dinucleotide, this hot spot, is repaired by cutting 20 nucleotides 5' and 4 nucleotides 3' to damage. Of course, this much more information can be gathered from this kind of map for UV damage. Most importantly, this method, nucleotide excision repair, also repairs the damage caused by very important anti-cancer drugs, cisplatin. And currently, we are generating a repair map for cisplatin-induced DNA damage, and we hope that that might have some implication for cancer treatment. To summarize then, nucleotide excision repair is initiated by dual incision, both in E. coli and humans. Excision is genetically controlled by UVR in E. coli, expedience in humans. And excision is not by endonuclease, exonuclease. It is by dual incision, removing 12 nucleotides in E. coli, 13 in humans, as shown here. And following excision, the repair gap is filled and ligated. And by capturing the excised oligomer, we have managed to generate a repair map for the entire human genome. I would like to close my presentation with a few words about cryptochrome and how the work on photolyze led to discovery of cryptochrome. Now, if you noticed in discussing photolyze and nucleotide excision repair, I referred to both E. coli and human nucleotide excision repair, but when I discussed photolyze, I mentioned only E. coli photolyze because humans do not have photolyase. This is a fact now, but for about 25 years after the discovery of photolyase, it was not a fact. It was a very controversial subject. A number of investigators reporting photolyase and others saying no. So in 1993, we decided to resolve this issue, and we carried out an extensive study on photolyase in humans, and we published a paper categorically stating humans do not have photolyase. And it didn't take long for the Human Genome Project to release their first draft and saying that humans do have a photolyase homologue. And so we, of course, were concerned that we made a major mistake and decided to work on this homologue. While doing that, we ended up discovering a second homologue. All of a sudden, we have not one, but two homologues. And we decided to investigate what these proteins do. As you see here at the primary sequence level, they are very similar to photolyase, and importantly, even at 3D structure level, they are very similar to photolyase. So we went on to purify them and test them for photolyase activity, and we found none. And at that point, we named them cryptochrome in analogy to blue light photoreceptors of that name in plants, and tried to see what they do. So they have high homology to photolyase. They are similar to photolyase. They have no photolyase activity. They're probably blue light photoreceptors. And this is what happened after these findings. This finding was made in the spring of 1996. And at the end of spring, in May, I made my annual pilgrimage to Turkey to visit my family and thinking about what we were going to do with those findings. And on my return trip, I read the Flight magazine talking about jet lag. and jet lag and circadian clock, and how this could be cured by blue light. And I came back to the lab and I said, cryptochromes are circadian clock proteins. And we wrote the paper at the end of August, and it was published in November. But saying is easy, proven takes time. It took us another 2 years to prove that these were circadian clock proteins. But just to say a few words about what circadian clock is, circadian clock is just like the clocks that we are familiar with. There are mechanical clock, electronic clock. They are based on either mechanic principles or electronic principle. And the circadian clock has same kind of design, except here the components are molecules. And their function is to keep time of day, to tell us what time of day it is. And that's what the circadian clock does. Circadian clock is an innate timekeeping mechanism, molecular mechanism, that maintains daily rhythmicity in biochemical, physiological, and behavioral functions. And this is independent of external stimuli. So, with this background then, to test whether the cryptochromes were circadian clock proteins or not, we generated mice with knockouts in cryptochrome 1, cryptochrome 2, and both cryptochrome 1, cryptochrome 2, and analyzed them for behavior. This is called actogram. You really don't need to know about the detail of it. We're recording the activity profile of mice that were kept for 28 hours for the experiment. For the first week, they were under light-dark cycle. The black traces indicate activity, and the white traces, regions show rest period. And mice are nocturnal. They run during the day, and they rest during the night. They rest during the day. And this is same for wild type, for cryptochrome 1 mutant, cryptochrome 2 mutant, and double mutant when they are under light-dark cycle. However, when you put the animals under dark condition, meaning no external input, then you see the difference. Both cryptochrome 1 and cryptochrome 2, without having to really know much about it, the behavior is quite different. But most importantly, the double knockout mutant has no circadian clock anymore. So this then established that these proteins are essential for circadian clock. And during, while this was going on, in the period of 1996 to 2000, there was a great deal of progress in the circadian field that led to the establishment of 4 genes, cryptochrome, period, clock, and BML1, as essential for controlling the circadian clock in humans. And this was the model that was developed. Clock, BML are transcriptional factors, activate transcription of cryptochrome and period. And these proteins come back and inhibit their own transcription, resulting in rise and fall of cryptochrome and period levels with periodicity of 24 hours. In addition, these proteins, meaning these core clock proteins, these 4 proteins, act on, in a given tissue, on 30% of other genes to confer this cyclic expression pattern with daily periodicity, and therefore control the biological function with daily periodicity. And among this, we found the excision repair protein, XPA, was also controlled by the circadian clock. And therefore, we asked the question whether the whole nucleotide excision repair is controlled by circadian clock. This is the circadian clock. And This is the nucleotide excision repair, and so we're asking a question if this clock controls this nucleotide excision repair with daily periodicity, and the answer is yes. In this panel, you show the expression pattern of XPA protein, which is essential for excision repair. Over the course of the day, it's low at 9 a.m. It goes up, reaching a maximum at 5 p.m., and then start going down to a minimum at 5 a.m. That then led to the prediction that mice would be more sensitive to UV light when exposed to at 5 a.m. when repair is low compared to 5 p.m. when repair is high. And we proceeded to do that, irradiated 2 groups of mice with ultraviolet light, one group at 5 p.m. when repair is high, one group at 5 a.m. when repair is low. And as you see, the 5 a.m. group at the irradiated at low repair period, low repair time, at 4 to 5 fold increase in invasive skin carcinoma compared to the 5 p.m. repair group. So currently, we are trying to find out or establish whether this periodicity also happens in humans, and whether that can be used in preventing, making public health recommendation to prevent skin cancer, and equally important, since this repair system works on cisplatin-induced DNA damage for cancer treatment, whether this periodicity could be used to improve cisplatin treatment of cancers. To summarize, to conclude, this is really a summary of my 40 years of work on photolysis, 35 years of work on excision repair in E. coli in humans, and over the last 20 years, we have found that the photolysed like cryptochrome links these 2 repair pathways that I have worked all my career. This doesn't really do justice to my colleagues, the acknowledgement that they have, as a large number of people have contributed to this work, and they will be properly acknowledged in the Nobel proceedings. And I have been very fortunate to have outstanding mentors, especially Stan Rupert, who has been my role model and who is the person who discovered DNA repair and introduced me to the DNA repair field. And being a Turk, I would also wish to acknowledge Muzaffer Aksoy, my internal medicine professor, who encouraged me to go to the United States and do science, and my other mentors, Dean Rupp and Paul Howard Flanders, my collaborators, among them Paul Modrich and others. And as I said, science is not done in vacuum. We have benefited greatly from colleagues who simultaneously carried similar work on photolysis, on circadian clock, and on excision repair, and I'm grateful to all of them as well, and I thank you very much for your patience.
Thank you very much. Now I think we can all sleep well at night, knowing that our molecules do the job for us. I would now like to welcome all three laureates up to the stage.
Keep the sound or keep it
I
down?
guess.